It's a pleasure for me to be here once again. I don't know how many of you were here 3.5 years ago when some of you, I gave the inaugural Cafe Scientifique talk, and we're going to cover some of the same material that I did then, but perhaps in a little bit more detail. And there'll be some slides that are a little bit technical, but we'll get through those very quickly, I promise, and we'll have plenty of time for discussion. Before I begin, I just do want to mention a few words about the history of this place. Some of you, Kevin just mentioned that it got started in about 1978. That happened to be the year I joined the faculty at Stanford. And I was, at the time, just before then, a postdoctoral fellow doing immunology research in a proper research laboratory at Stanford. That was focusing on fundamental studies of the immune system, and of course that could only be done in studies of rodents, specifically of the mouse. And my mentor at the time, who was a very famous professor, felt strongly that the only way to gain meaningful understanding of the immune system was to study mice. And the reasons for that were that they can be made into genetically pure clones, if you will, and there were lots of reagents that were available to study the different types of white blood cells and other elements of the immune system. And when it came to studies of humans and the human immune system, he felt very strongly at the time that that was a complete waste of time. Even though in the end, as a physician, he knew that that was the ultimate goal, which was to understand the human immune system and then eventually be able to take advantage of that understanding in the discovery and development of new treatments. So when I was offered a faculty position in the Department of Pathology here at Stanford, and also offered the idea, or at least the concept of creating and establishing a blood bank, I thought it was a great idea, and he didn't. In fact, when he first heard about this, he said, "You cannot take this job. It would create we were going to compete with one another, and I don't think that's a good idea." All of his former trainees actually left the university and went to other universities. But when I told him that I was actually going to change out, as it were, and start to study the human immune system, he first said, "Well, you're going to waste your time again." And the second thing he said, "Well, that's fine. That's fine. There won't be any competition." But be that as it may, in addition to wanting to help a noble cause, which was to establish a blood center that could procure blood products and services for Stanford, mainly for Stanford and Children's Hospital at Stanford, I knew that the only way to make headway in understanding how the human immune system works was to have a blood bank. And the reason for that is that while most of the components of blood, the red blood cells, the platelets, and the plasma are very helpful and save lives every day, the white blood cells are actually removed from your donated blood. And the reason for that is because white blood cells include cells that routinely attack any tissue or any substance that they don't think belongs there. And as I'll mention in a few moments, some elements of the immune system are trained from the time of early embryogenesis to avoid attacking self, while at the same time universally attacking anything that is not self. So anything that is not self is anything that is in anybody else. And so as a result, we try to get rid of the white blood cells from donated blood before they're transfused, lest those cells attack the recipient of those transfusions. Now, ordinarily, in healthy people, the presence of those foreign white blood cells is harmless. But in patients who have received high doses of chemotherapy or have gotten transplanted organs and whose immune systems are depleted, those white blood cells can in fact be very harmful and very dangerous, even cause death. So, as a consequence of that, from the time I started here, and when we started, by the way, I think there were 3 employees. Today we have over 300. And we were not in this building, we were elsewhere. But from the time we started here, we have routinely removed the white blood cells and collected them and made them available for research. Not just my research, but the research of others, both inside Stanford University and all around the world. And it's been an extraordinary resource that has helped in the kind of work that we've done and the kind of work that continues to get done. So with that preview, I'll get going on what you're here to hear. And that has to do with the development, the discovery and the development of cancer vaccines, and in particular, therapeutic cancer vaccines that involve these rare white blood cells called dendritic cells. Now, before I get into it and tell you about the clinical data, I have to give you a little bit of a primer on the immune system. And I apologize to those of you, like members of my lab, my research lab, that are standing in the back of the room, and others who are immunologically educated and sophisticated, but I got to go through this for those of you that are not that familiar with the immune system. So this is the way I see the immune system, and I know it's an oversimplification. So the immune system is our major defense against viruses, tumors, and other, quote, foreign invaders that shouldn't be there. The immune system uses its white blood cells and the products of those cells. Some of those we call cytokines, inflammatory mediators, antibodies, to combat invaders, to these invaders. Now, in point of fact, the immune system consists more than just white blood cells. Your skin is part of your immune system because your skin is also a protective element. But for our purposes tonight, the immune system consists of white blood cells. Now, among the white blood cells, there are many different types of white blood cells, some of which have been appreciated only in the last decade or so as we've had the ability to begin to decipher all the different types of white blood cells. You can consider those kind of different components of your army, your navy, and so forth, different types of soldiers, if you will. One way of looking at the immune system is to consider it in two basic arms, the innate arm and the adaptive arm. The innate arm consists of cells like granulocytes, also known as white polymorphonuclear leukocytes, macrophages, natural killer cells, mast cells. These are white blood cells that can react extremely rapidly when something foreign enters the environment, but they do so with limited or no specificity. So they basically are like bombs that explode, attempting to gobble up or kill anything in the environment. Now contrast that with the adaptive immune system, or the adaptive immune cells that are comprised mainly of T lymphocytes, or T cells, and B lymphocytes, or B cells. Because these cells can recognize and react to foreign substances that are known as antigens on the basis of their molecular features. So in other words, each of these cells has receptors on their surface that are geared to recognize specific molecular structures in the environment. And if they encounter those structures, they get very excited, and they proliferate, and they reproduce, and they go into action. And these cells provide long-term antigen-specific protection. Sometimes we call that immunological memory, immunologic memory. And it is these cells that are the key to vaccination. Because when you get vaccinated, most of the time, your vaccines are used to prevent serious infections, not to treat, but to prevent. And the way that happens is we introduce into your bodies a substance that either is a component of a dangerous virus or something that looks like a dangerous virus. And your immune system then is your adaptive immune cells, your T cells and your B cells will then react to it. But more importantly, they build up a memory such that if you ever encounter the real thing later in life, you will react very swiftly and very powerfully and presumably prevent that dangerous virus from getting a foothold. And this works most of the time. And immunologic memory can last many years. Okay, now what about dendritic cells? Well, it happens that dendritic cells represent the link or the bridge between the innate and the adaptive immune systems. How? Because dendritic cells are your sentinels. They reside in virtually every tissue, and they're constantly on the lookout for foreign substances, things that shouldn't be there that represent a danger. And if they encounter those substances, then they take them up and they will, in fact, introduce them to both the innate and the adaptive immune cells. So they're the initiating cells, and they're critical for activating both arms of the immune system. Okay, now, tumors in the immune system. Tumors represent a unique and very difficult challenge because whether we like it or not, the vast majority of tumors consists almost exclusively of self. They're not, most of them, there are a few exceptions, they're not caused or containing viruses or other foreign pathogens. In fact, they're made up almost entirely of the same substances, the same antigens, the same components as normal tissues. That makes it very difficult for the immune system to recognize them because they're not really foreign. And as I said before, the adaptive immune cells are trained from early life to avoid attacking self. And there's a variety of interesting molecular mechanisms that are used to do this, but I won't go into that now. But suffice it to say, the goal of immune therapy or therapeutic vaccination is to overcome the reluctance of the immune system to attack the tumor or to produce tumor-specific antibodies or tumor-specific white blood cells outside of the body and inject them into patients to attack the tumor. We call the latter passive immunotherapy because the patient's immune system doesn't have to do any work, and I'll call the former active immunotherapy because we're trying to induce the patient's immune system to attack the patient's own tumor. Does that make sense? Okay, good. So where are we in regards to tumor immunotherapy? What are the various approaches that are now in use or in experimental development? Well, on the left, we have a list of passive immunotherapeutic approaches. The one that is the most important today are monoclonal antibodies. Now, if I were to give this talk 20 years ago, and I did give lectures 20 years ago, believe it or not, I could only talk about monoclonal antibodies as hopeful, futuristic therapeutics. In fact, I edited a whole book on the subject about 27, 28 years ago, where a number of researchers who were investigating how to make monoclonal antibodies wrote chapters, and we all talked about how the future would involve monoclonal antibodies primarily for the treatment of cancer. But it took a lot longer than we thought to finally see the day when monoclonal antibodies could be made in a manner that was clinically useful. But they are now, and many of you have heard of them because they are now standard parts of treatment for many cancers. And they have certain advantages that I'll talk about in a moment. There are other ways of passive immunotherapy. You can imagine using a patient's white blood cells that have been removed and trained and expanded and then given back. This is all experimental, and I'm not going to talk about those tonight because they're highly experimental and not close to FDA approval. Now, on the right, we have active immunotherapy, where we're trying to induce the patient's immune system, the patient who's bearing the tumor, to attack the tumor. And one approach is to use these dendritic cells obtained from the patient and then armed with an antigen from the tumor and then introduced back into the patient in an effort to use those cells to activate the patient's own immune system. And that's what kind of work we've been doing for the past 20 years. But related work involves using different types of vaccination, like various kinds of vectors or safe viruses to deliver those antigens to dendritic cells and induce the dendritic cells in the body to become activated. And then another approach that's been in the news lately, a monoclonal antibody that's not directed against tumors, but actually directed against T lymphocytes that actually causes those T lymphocytes to become activated. So those are the kinds of approaches that are now in development as active immunotherapeutics for cancer. Now, just a few words about monoclonal antibodies, which is not the main topic for tonight, but since these antibodies are already part of standard care, I think it's important to at least acknowledge their use and their importance. So, a number of antibodies, which are proteins that are produced by your B cells, by the way, are now used therapeutically. And the key to their ability or their access and their ultimate commercial success was a breakthrough by investigators, many of whom were at Genentech years ago. in how to produce these antibodies in massive quantities. Because each monoclonal antibody is a single antibody. Now we have in our blood and throughout our bodies probably 1 million different antibodies, all at very low concentration. But monoclonal antibodies refers to antibodies that have been produced as a homogeneous single antibody with a single target in mind. So for example, Avastin is a monoclonal antibody that recognizes a blood growth factor. The anti-HER2/neu is another antibody that recognizes an antigen on breast cancer, tumor cells, and so forth. And the way these antibodies are produced is using recombinant DNA, and they are now available. I think there's about a dozen such antibodies that have been approved for the treatment of various cancers. And they're generally very safe and well tolerated because they're so specific. And that's the value of the immune system, or the power of the immune system, is that in contrast to most chemotherapies or standard chemotherapies that cause considerable collateral damage because they're non-specific, they simply kill cells that are growing fast, for example. Products of the immune system, be they cells or antibodies, are very specific for their targets, and they generally don't cause much in the way of collateral damage. So these monoclonal antibodies are very useful. They recognize only a single target, but they must be administered repeatedly over time, and in general, while they're safe, they're only partly effective. It's very, very rare when you get a cure from a monoclonal antibody, almost never. Okay, so let's go back to the concept of therapeutic vaccination or active immunotherapy. I told you before that most vaccines are used for preventing diseases. In fact, there is one vaccine that's been approved today for preventing cancer. Do you know which one that is? It's an approved vaccine for preventing cervical cancer, right? Because it's directed against a virus called the human papillomavirus, which is actually responsible for causing cervical cancer. And it's one of those very rare human cancers that is known to be caused by a virus. And so it's possible to prevent such a cancer by simply preventing infection with the virus using a classical vaccination approach, and indeed it works, and it's widely available and safe. But what I'm talking about is using a vaccine strategy for patients who already have cancer. And so, as I said before, the old view was that the inability of the immune system to recognize and attack tumors is inherent and irreversible. And that view was based on the pervasive view that because the white blood cells, particularly the T cells during their development, are trained to avoid killing self or attacking self, how could we ever turn the patient's immune system against a self-tumor? The new view, and the one that I hope was right some 20 years ago when we had this concept, was that in fact that's not true, and that in fact that one can induce effective anti-tumor immunity in a tumor-bearing host, provided that the right antigens, the right tumor substances, are presented to the adaptive immune system by the right antigen-presenting cells, dendritic cells, and that the immunosuppressive environment in cancer patients can be neutralized. So that was the hypothesis. We know today that that is in fact correct. So let's turn to dendritic cells, and I'm going to give you a perspective that is somewhat historical, based on what we knew when we started, which wasn't a lot, and what we've learned since. So dendritic cells are white blood cells. They're quite rare. They represent about 1 in 100 of the so-called mononuclear white blood cells, or 1 in about 500 of all the white blood cells in blood. And what they do, particularly the classical dendritic cells, is that they capture, process, and present antigens in the environment mainly to T lymphocytes. They also activate innate immune cells, such as NK cells, and they can also activate B cells. Now, there's another type of dendritic cell called the plasmacytoid dendritic cell, about which I won't speak much tonight, except to tell you that this cell type is the main source of type 1 interferon. And interferon is a very important substance that helps fight against viruses. But this cell doesn't participate, we don't believe, in the kind of vaccination that we're going to talk about. So this is what dendritic cells look like under the microscope, and you can see why they're called dendritic cells. It's because they have these long dendritic arms, dendrites, if you will. It's interesting because when they were first noticed, it was assumed that they were part of the nervous system because nerve cells look just like this. But in fact, these are not part of the nerve system. They come from bone marrow originally, and they're white blood cells. And you can see that they occupy almost every tissue you look for. They're big cells. And here's their life history. I apologize again, this gets a little technical, but on the left there, you can see that the progenitors or the precursors of dendritic cells are created in the bone marrow. They then travel via the bloodstream to wherever peripheral tissue they're going to ultimately occupy. And I'm going to show you an example in the skin. So in the superficial layer of the skin, which is called the epidermis, we have dendritic cells called Langerhans cells, and then in the deeper layers we have dermal dendritic cells, but they both come from the bone marrow. And they'll stay in the skin for your entire life, unless, and they sample the environment during that entire time, unless something comes along that shouldn't be there. We can call these pathogen-associated molecular patterns or danger signals, because dendritic cells have on their surface and inside them receptors that can recognize substances that aren't normally present, that are foreign. And if they interact with those substances, they get very excited, they change their morphology, they get bigger, they engulf that substance, they bring it in and they process it, and they leave. They leave their posts for the only time they will do so in their entire life, and they leave via the lymphatic vessels, and they travel into the draining lymph nodes. We have lymph nodes all over our body, and they end up in those draining lymph nodes where they will then present the antigen that has been carefully packaged for recognition by various types of T cells, CD4 T cells, CD8 T cells, natural killer cells, and B cells. And the result of that is the beginning of an immune response. And this is how we react immunologically to anything that comes along that shouldn't be there. So for our lab, and for those of us that study these cells, the dendritic cells is the center of our universe, or immuniverse, and they're incredibly important because without these cells, we wouldn't live. Okay, so going back in history, in 1988 or so, the blood center was, I guess, about 9 or 10 years old at that time, and much smaller than it is today. But I had a couple of new postdoctoral fellows in my lab, and by that time I had become very interested in the possibility that there existed human dendritic cells, and might they be important. There was some work in the mouse that suggested such cells existed and were very important, but little or no work had been done on human dendritic cells. And what I suggested to these new students, new trainees, is that they develop a way of isolating these cells so that we could study them. And indeed they did, and they found out that what they looked like, they studied their behavior, they showed that, remarkably, that if we isolated those cells and then cultured them outside of the body in test tubes together with T cells and a new antigen that these people had never before seen, that the donors of those cells had never before seen, we could actually vaccinate those T cells in a test tube. We could actually sensitize T cells that had never before seen this antigen, provided that we introduced the antigen to them with dendritic cells. And we published that information in the early 90s. And that led to the idea that, gee, if we can actually vaccinate the lymphocytes in a test tube, maybe we can also vaccinate the lymphocytes in patients. And so that led to the idea of using dendritic cells as therapeutic vaccines. And that idea, I had this idea, which is actually a pretty simple-minded idea, to be honest with you, back then. And I did what every good Stanford employee does. When he has a good idea, he goes to the Office of Technology Licensing and says, I've got this idea, it might be valuable, and maybe you should file a patent, Stanford. And indeed, they did that. And we continued to do our studies. And we came up with this rather simple approach of trying to treat cancer. And the goal was to induce anti-cancer or antiviral immunity using dendritic cells loaded with tumor antigens. Now the word autologous, that big word there, means from the patient himself or herself, as opposed to from somebody else. And then the methods were to generate or isolate these cells from the blood, load them with antigens from their tumor, and then return them. That simple. So I thought that this approach would be very safe because, in contrast to blood transfusions, where we take blood that has lots of different cell types, including a few white blood cells, and we transfuse an unrelated patient, all we were doing was isolating a rare type of cell from the patient himself or herself and returning them 3 days later. So I thought this would not present any harm. I didn't go to the FDA at that time because there was no reason to, at least I didn't think so. I did go to the committee at Stanford, the experimental approval committee called the Institutional Review Board, and they approved it. And then well, let me just tell you that in the years subsequently, in the next 10 to 15 years, we used this approach in small experimental pilot clinical trials to treat malignant lymphoma, multiple myeloma, prostate cancer, HIV infection, and colorectal cancer. And I'll show you a couple of highlights. But the first trial that we undertook was in malignant lymphoma. And these were patients that had what are defined as low-grade non-Hodgkin's B-cell lymphoma. Low-grade doesn't mean not serious, because it just means slow-growing, but all these patients were dying of their disease. And I approached the chief of oncology at Stanford, whose name is Ron Levy, and Ron took care of these patients with lymphoma and was also interested in developing potential immunotherapeutic strategies. And I suggested that perhaps we could work together, that we would isolate the dendritic cells if he could provide us with the tumor antigen. And he agreed, and we subsequently then developed a strategy, the first strategy for using these cells clinically. And it's illustrated on this slide, where this is a hypothetical patient. It's shown there as a male, but it could be a male or a female, obviously. The patient's tumor was biopsied, antigens from the tumor were obtained, and we, in the meantime, had the patient come to the blood bank where he or she underwent a outpatient 1-hour leukapheresis procedure. Does everybody know what a leukapheresis procedure is? Some yeses. It's basically like a blood donation, only it's done with a little machine, but it's pretty painless. It takes about an hour. So we then isolated lots of white blood cells, and from those white blood cells we purified the dendritic cells. We cultured the dendritic cells with the antigen over a couple of days, and then we washed the cells and injected them back into the patients. And that was, as I said, that was back in, I think we started that in 1994. And what we found is that among the first 10 patients, all of whom had failed all therapies and had large growing tumors all over their bodies, is that most of them responded to this vaccine immunologically. That was important, that we could actually induce an immune response. More important was that 2 out of the first 10 experienced complete remissions. That's pretty amazing. And then a couple of other patients had what we call partial responses. Now, it's important to point out that while not every patient responded favorably, actually inducing a complete clinical response where the tumor literally disappears is actually pretty exciting, especially when you're dealing with tumors that were otherwise untreatable. So we were very excited, and we published a paper in the journal Nature Medicine in January of 1996. And that was the first description of this whole approach. I think we only had 4 or 5 patients in that paper, as I recall. But we went on and treated some more patients, and we got very encouraging results. And all of that was subsequently published. And once that paper was published, then the excitement started to grow, and we ourselves got more excited. But I was a little bit concerned that one of the limitations that I had was our inability to get a lot of dendritic cells. I thought maybe one way to make the vaccine even more potent would be to try to capture even more dendritic cells. And when we did these leukapheresis procedures, we're only getting 2 or 3 million cells, which is a tiny number of cells when you realize that there may be 10 to the 12th white blood cells in the body. So having a couple of million dendritic cells seemed to me to be maybe too few. So I read some interesting papers about the about an interesting molecule called FLT3 ligand, which was a protein that acts on the bone marrow to make a lot more dendritic cells. And a biotech company called Immunex, located in Seattle at the time, was producing this molecule using recombinant DNA techniques. And so I journeyed up to Seattle and asked if they would be willing to give us some of that protein so that we could use it to treat our patients and make more dendritic cells. And after a long and very painful negotiation, they agreed to give us enough material to treat 12 patients. And we decided to treat 12 patients with metastatic colorectal cancer, mainly because this was also an impossible disease to treat, or thought to be so. And we treated 12 of the patients. Now, I don't want to get overly technical, but we used a single small 9 amino acid peptide as our antigen from carcinoma embryonic antigen, which is an antigen that is overproduced by a large percentage of patients with this tumor. The tumor actually produces this stuff. So we thought it might be a good antigen. And the way we carried out this experiment was to treat the patients with FLT3 ligand for about 10 days in order to generate lots of dendritic cells, which we then would capture with our leukapheresis procedure. And then we would load those cells or arm those cells with the antigen and return them to the patients. We did this twice. So we treated them with the FLT3 ligand, captured the dendritic cells, and then we stored some cells and gave them again a month later. So there were 2 treatments. What we found is that this material worked very well, and we got about 100 times more dendritic cells. That was good news. We also had some pretty miraculous results in that we had a couple of patients in that tiny little trial that had dramatic tumor responses. So this is one such patient where on the left, before treatment, this patient had metastasis to the lungs, shown here in the arrow. And then after vaccination, the tumors disappeared. Very exciting. We found of the 12 patients that were treated in that tiny trial, about 5 or 6 of them had meaningful clinical responses. And this is a technical table for which I apologize, but what it's intended to convey is that the patients who responded clinically were the same patients who responded immunologically. And that's actually a big deal, because we could actually count in the blood how many antigen-specific T cells were produced as a result of the vaccine. And the patients who responded clinically, where their tumors shrank, were the same patients who had an expansion of the antigen-specific T cells, proving that the vaccine was working through an immunological mechanism, which is good news. So I think I'll skip over that and jump immediately into the limitations. So we were very excited by these types of results. Once we published that first paper in early 1996, researchers around the world started doing their own dendritic cell clinical research. And sadly, they all decided to use a different approach. They thought it was great, but they thought maybe they could have a better mousetrap. And the results were that the trials were very variable. Some worked, some didn't. It was unclear. And some of the reasons for those problems involved which tumors do you treat, what are the best tumor antigens to use, what's the best method for isolating and activating the dendritic cells, what's the best way to inject them. And of course, the cost and complexity were high, because when we started, we didn't need FDA approval. But after we got started and we started publishing, the FDA in its wisdom decided, well, that's something we should regulate. This is true. So they became very concerned that if we were isolating cells from patients and manipulating them in vitro, that there was maybe some harm could come. Maybe we would accidentally introduce some dangerous bacteria, for example, or something else. And so I don't want to be too cynical about this, but suffice it to say that when we did the first experiments, we were able to treat each patient for less than $1,000. All in, all in. If I were to try to do those same experiments today, it would cost me about $40,000. And it's all because of the requirements placed upon us by the FDA. It's not evil. It's not that there's any evil intent. It's just that they want to guarantee that the product that we would be injecting into patients are sterile, guaranteed sterile, and guaranteed to meet the specifications that they have given us permission to study. But I can tell you that puts a real dent in the ability to do this kind of research. So obviously in the long term, in point of fact, I don't think long term this is a viable approach. It's simply too expensive. But on the short term, it demonstrates the proof of concept, which is so important. And we were, again, quite excited. Now I want to go back and mention to you that I went to the Office of Technology Licensing in 1991 or 92 with this idea of using dendritic cells as cancer vaccines. The Office of Technology Licensing at Stanford then is empowered to try to not only file patents, but to try to find companies, existing companies that would be interested in working to develop these approaches as approvable therapeutics. I knew that given the FDA's interest in regulating this approach, that any hope of developing it as a viable therapeutic would require a very large amount of money, much more than we could ever get in the form of research grants, and that it would require a corporate effort. So did Stanford. However, there were no existing pharmaceutical companies in the early to mid-90s that had any interest in this approach. And the reasons should be obvious to you, because it's really expensive. And every time you want to make a vaccine, you have to make it from scratch from the patient. So we call this a customized or patient-specific therapy. In contrast to a drug, where you use the same drug, or almost the same drug, for every patient with a given cancer. That's a lot more efficient from a manufacturing perspective and much more profitable for the companies. So this wasn't easy. So when it became apparent that we weren't going to find someone to license the technology, I, together with a colleague of mine, a faculty colleague, Sam Strober, went about to try to see if we could convince investors to create a company to develop this strategy. And we succeeded eventually. And that company was Dendrion, or it's known as Dendrion today, and some of you might have heard of it. It's certainly been in the news a lot. And Dendrion, in its wisdom, hired a number of my former trainees, my former postdocs, some of whom are really smart people, and they went to the company as scientists, and I worked very hard to advise the company on what their strategy should be. And the first tumor that they decided to pursue was metastatic prostate cancer. Now, why metastatic prostate cancer? There's a good couple of reasons for it. Many tumors occur in vital organs, like lung cancer, or even colon cancer, or pancreatic cancer. These are all organs that we need to live. It happens that the prostate is not particularly important. And if we happen to destroy it through an immunologic attack, that's okay. One way to look at this whole vaccine strategy is we're trying to create an autoimmune disease. If you think about it, what happens in autoimmune disease? Basically, your immune system turns on your normal tissues and creates an inflammatory response that ultimately results in the destruction of that tissue. That's what happens in rheumatoid arthritis, in multiple sclerosis, in systemic lupus, and so forth. So what we're trying to do is create such an autoimmune disease, but we want it to be very specific. And so one example of an organ that we thought would be a reasonable target for such an experiment, a big experiment, not just 12 patients, was prostate cancer. In addition, of course, it's very common in men. And there was no treatment for the disease once it became metastatic. Essentially no treatment. So for those reasons, the company decided to pursue prostate cancer. They followed the same basic strategy that we had discovered here at Stanford, here at the Stanford Blood Center, and developed a vaccine antigen that consisted of a prostate-specific antigen, not PSA, as some of you know, that's used for the PSA test, but an antigen called prostate acid phosphatase that is a protein that's expressed by the normal prostate as well as by about 95% of prostate cancers. And they use that antigen together with another component called GM-CSF, which I won't torture you with right now. And they went about to do clinical studies in patients with hormone-resistant or castrate-resistant advanced prostate cancer. Well, when we started doing that work, when the company started doing that work in the mid-90s, I thought it would take at least 3 or 4 years before it could possibly finish up and get approval. Let me point that out to you again. I said I thought it would take at least 3 or 4 years. That was in the mid-90s. So 15 years later, in April of 2010, they finally made it over the finish line as the first active immunotherapy, the first therapeutic vaccine to be approved by the FDA. The FDA was not gentle and easygoing when it came to approving this approach. We can go through a little bit about what was involved there, but before you hear on this slide are the results from one of the 3 clinical trials, 3 sort of late-stage clinical trials that the company performed. This was not their last trial, this was their 2nd trial. And what it shows you in red are the patients who received the actual vaccine, and in blue, the patients who received a placebo, a partial vaccine that did not include the antigen. And on the y-axis there is survival. So what you can see is that the vaccinated patients lived longer than the control patients. The patients were otherwise identically treated. Now, this doesn't show you, so what's not shown is that these patients are getting cured. They're not getting cured for the most part, but they're living longer. And in this particular study, was an average of about 4.5 months longer. Not an average, I should say, a median. By the way, there's a big difference between mean and median, and I just want to point that out to you because this is a very controversial point, because the question is asked, well, gee, how valuable is an extra 4 or 5 months of life? Well, that's the median. That means that half of those patients get less benefit and half of them get more benefit. So some patients will live a year or years longer as a result of receiving this vaccine. But beyond the fact that the vaccine demonstrated efficacy, in point of fact, it was very safe. So the name of this vaccine, this kind of silly commercial name, is called Provenge. I guess it's easy to remember. And in April of 2009, the company announced the results of their last trial, their big 500-patient trial in patients. And it really was the first convincing demonstration that an active immunotherapy with a tumor antigen-based vaccine can provide real clinical benefit. The vaccine was well tolerated. I think only 1 or 2% of the treated patients ended up with any serious toxicity. All of it was transient, none of it permanent or long-lasting. And a year later, the vaccine was finally approved by the FDA. It wasn't easy, as I mentioned before, because the company thought that they were going to get their vaccine approved about 3 or 4 years earlier, and they submitted their application for approval, as some of you may know. And then the FDA organized an independent panel of experts to review the data, after which they recommended that the FDA approve the vaccine. But a month or so later, the FDA said no and wanted the company to carry out an additional trial, which they did. And it took several years to complete and over $100 million, I believe. But they ultimately finished their vaccine trial, and this is what the result was. So that's very gratifying, very exciting. But as I mentioned to you, and as is constantly in the news these days, it's pretty expensive. And this cartoon sort of illustrates one extreme view of this particular vaccine and other very expensive medicines. And it says this is one of those. This is a patient who is going to get an expensive medicine, and her doctor, or this could be the pharmacist, I'm not sure, is saying this is one of those new miracle drugs. If you can afford it, it's a miracle. Dendritic doesn't want to hear about that, I'm sure. But if you follow the news, because this company is just, everybody loves to talk about it. not just me. And there are those that are very negative and those that are very positive. And there's a lot of talk about how costly it's going to be, because the company announced some time ago that they were going to charge $90,000+ per treatment. It's only approved for treatment. By the way, it's given 3 times, 3 injections at 2-week intervals, and they were going to charge $90,000 for that. And that sent all kinds of shock waves and the like, because it sounded like such an enormous number. And I don't want to be viewed as a defender of the company. I don't have a relationship with the company anymore. It's been about 10 years since I've had one, although I still own a few shares. But actually, $90,000 is not so incredibly expensive compared to some of the other expensive biological drugs that are used to treat cancer. But rather than defend it, I will simply say that, as I've experienced here at Stanford Blood Center, it's just incredibly expensive to produce this particular vaccine. And I think it's the best part of what has been accomplished is to demonstrate that this approach can work. It's not yet to the point of curing patients, but it can work and it's safe. And it's the first one. So what does that mean for those of us that are in the field? It means that there are now 100 or 1,000 different investigators dreaming up new ways to do this better and cheaper. And I personally know of at least 15 different vaccine strategies that are in development today as a result of Dendrion's approval. So that's the real importance of this, at least from my perspective. So, so what's the future? Well, with regard to the next generation of dendritic cell therapy, I think you'll probably agree with me, or at least I'll try to convince you, that the best approach, the most practical approach, is to avoid actually isolating these cells and manipulating them outside of the body, but rather figure out a way of targeting the cells inside the patient's body and activating them and arming them with the right antigens. And even though that might sound like a daunting task, in point of fact, it's feasible. And we and other investigators are in fact evaluating those approaches. And unfortunately, we have to use mice for that. It's interesting, I go back and I say, gee, in order to do this, we have to use mouse models because it's too expensive to do it in humans right now. But in fact, there are approaches, and I've listed some of them here, and I don't think I'll spend time talking about them because it's a little technical, other than to tell you that we really can do this. And I believe that we're not very far off from seeing these types of approaches evaluated in the clinic. So the future is bright for immunotherapy. I talked about monoclonal antibodies before. There are going to be a lot more monoclonal antibodies in the future. Genentech isn't going to go away, although it's now owned by Roche, right? But there will be many monoclonal antibodies there, and many more. They're safe and they're effective. They're just not generally curative. And there will be drugs that target and activate dendritic cells. I think we'll see that over the next several years. And there will be drugs that target and activate other types of white blood cells. I mentioned one drug in late-stage development right now, anti-CTLA-4 antibody, which targets and activates T cells. So that closes my formal remarks. I just want to acknowledge that this is work carried out over the last 20 years. I haven't listed anything like all the people who were involved on this slide, but some of them are listed here that were involved in the initial work in isolating human dendritic cells and developing those methods. The lymphoma studies that were done in collaboration with the Levy Lab, the colon cancer trials that were done somewhat more recently. It's great to look at this list and know that many of these former trainees and postdocs are now professors at other universities. There's also one person who's looking over there, Claudia Benecke, who's actually been with me and working in my lab and actually supervising the lab since I started some years ago. And so it's nice to see Claudia here because Claudia was the one who actually did the initial dendritic cell isolations for the patients. So she did all of those isolations here in our building. Well, it wasn't this building, it was the previous building. And that was before the FDA said you can't do that in your building. You have to have a special building for that. But thank you, Claudia. And then, of course, the Stanford Blood Center and its dedicated staff. I want to make clear that this kind of research would have been impossible in virtually any other place on the planet, as far as I know, because the fact is that this blood center is unique. It's part of a university. It's inside the medical school, in fact, inside the Department of Pathology. And when we started the blood center some 30 years ago, we had a dual mission. The first mission was to obtain, provide the best possible blood products and related services to patients. The second, but almost equally important mission was to make sure that the blood center was a training and research center as well, where we can train the next generation of scientists and physicians and educate them with regard to immunology and hematology. And believe it or not, it's actually fulfilled its mission over the last 30+ years. And so that was one of the reasons why I wanted to do it or get it started, and it actually has turned out to fulfill that particular dream. I hope it can continue. It's not easy, but without the cooperation of the entire blood bank staff, which includes its nurses, the administrators, our own research group, and of course the blood donors who make it possible, the work could not have been done. So here's a photograph. I try to do this every year. I don't, but I try. Of the lab as it was about a year ago. These are the members of my research group, some of whom are still in the lab. And you can see Claudia there next to me, in case you didn't see her way up there. She still looks the same. And then a number of both postdocs and graduate students, some of whom are still here. So that's how it was all possible. And I think I'll stop there, and then we can have discussion or questions, if you have any. Thank you. Okay. Yes, ma'am.
Is this vaccine individualized then?
Yes, this vaccine is individualized. So you're talking about the Provenge vaccine for prostate cancer? Yeah. Every patient who is to be treated with that vaccine actually undergoes a leukapheresis procedure, and then the cells are sent to a manufacturing facility or a special facility where the dendritic cells are enriched, and they're then pulsed or cultured with this antigen from the tumor, and then washed, and the cells are then returned, sent back where they're injected into the patient, and this is done 3 times over a 6-week period. Yes, sir.
So for the tumor, is it a specific tumor that you're working on, or is it generic tumors?
That's a great question. The question was, does this vaccine work only for the specific tumor, or is it a generic approach for other tumors? Well, the answer is no and yes. It's the Provenge vaccine is only for prostate cancer because it's not the cells. The dendritic cells or potentially can induce an immune response against any antigen or any tumor. But if you arm those cells with a prostate cancer-specific antigen, they're going to induce an immune response against that antigen, against those tumors. Conceptually, you could do this for any tumor. So you could do it for breast cancer, colon cancer. Indeed, the company, Dendrion, did have programs begun years ago when I was involved with them in other tumors, but eventually it became obvious that they could only afford to do one. But at this point, maybe they can afford to go back and do the others. But each tumor then would require a separate antigen with the same cells. And of course, separate clinical trials that would take a long time and require FDA approval. Yes. By the way, I'm not opposed to the FDA. I know this is going to end up on YouTube, and I don't want to suggest to you that the FDA is evil inherently. It's just that, and they serve a very important purpose, but in this particular case, I think that the regulations are extreme, and they've just made it almost impossible to do research of the type that we were able to do. That's how my feeling. We can talk about that more, but yes, ma'am.
Are insurance companies approving payment for this treatment, and do you know of any trials or something that will be in the near future for breast cancer?
Okay, let's start with the first question. It gets back to the issue of the cost. The question was, are insurance companies paying for this vaccine? Well, that's in the news today too. In fact, it's really interesting to follow this because you can get a real education on how healthcare is paid for. And in fact, one usually assumes that once a new treatment is approved by the FDA that it's automatically paid for. Well, yes and no. And so in fact, there is a special arm of Medicare. Most of the patients who have prostate cancer would be eligible for Medicare, so Medicare would be the principal source of reimbursement. And Medicare is taking a year to evaluate this. I think in the end they will reimburse, but I think probably they're going to be very careful about making sure that the only patients who are reimbursable are those that have the exact severity of tumor that were treated in the trials, which is patients with advanced hormone or castrate-resistant prostate cancer and minimal or little in the way of symptoms. That was the definition of the patients that were in the trials, and those will be the patients who are eligible for reimbursement. So, but I would guess it would be very shocking if this wasn't reimbursed. It would be the first time, but so I suspect it will happen, and the other private insurance companies will undoubtedly follow along. Yes, Walter?
Would it be possible to select patients that might respond to other patients? Pre-select them?
Yeah, wouldn't that be great? The question was, is it possible to identify patients who would be potentially better responders and other patients who may not be responders at all? And that would make, of course, a much more efficient and effective therapy. I don't know the answer to the question. I think one way of looking at it is that, you know, when we do cancer clinical trials, we always do those trials in the sickest patients, the ones with the most advanced disease, whose options are limited. And data from, most data, including data from mice and humans, would suggest that the best responders would be the patients with the earlier disease, where the tumor burden is less and the immunosuppression is less and the patients are generally in better condition. But in general, once a product is developed and it's approved for the very specific indication, that's the indication it will be used for. Now, my guess would be that over time, efforts will be made to evaluate, to answer your question, basically, to evaluate the efficacy of the vaccine in patients that are either earlier stage or have particular types of prostate cancer or not. But as I said before, I think the wave of the future is going to be coming up with drugs that can actually target the cells in vivo and not manipulate them in vivo, ex vivo. Yes?
What types of SAEs are reported with use of the drug?
I'm sorry?
What types of SAEs
Oh.
are reported with the use of the drug?
Yeah. So the question was, what types of SAEs, which are severe adverse events, for those of you that don't know about SAEs, as opposed to SAT scores. This is SAEs. And in fact, one of the remarkable aspects of these immunotherapies, particularly the dendritic cell, the Provenge product, is that there were very, very few SAEs. What typically occurs when the patient receives these dendritic cells is that they often will get chills or fever, and that can last for hours to a day or two, and then it goes away. In the trials, there was a slight increase in the incidence of stroke. That sounds really bad, I know. But when the statisticians looked at all the data, they decided it was meaningless. But I mention that because it was kind of a curiosity. I don't know why that would be. But for the most part, I think it's fair to say that there were very, very few severe reactions. To my knowledge, no one died as a consequence of receiving this type of vaccine. And as I said, most of the side effects are mild and they go away. It's very different than, you know, there was a very interesting article that was in today's, on the internet today. I have to mention it because I was wanting to make sure that I had read up and didn't make any mistakes, because I'm not a representative of Dendrion, and as I told you, and I didn't want to say anything that was not accurate. And there was an article today, and it was by a guy by the name of Clark, I believe, or Miller. Anyway, it was in something called Minionville. If you look up, go to Dendrion, and then you'll see the news, you'll see that this article, and it was actually a remarkable article. I thought it was really interesting and well written. And it was all about the issue of the cost relative to the cost of chemotherapy. Because the paper that described the last trial that Dendrion did was published just 3 days ago in the New England Journal of Medicine, which is the most prestigious of our medical journals. And in that particular journal, there was an editorial written by a scientist from the NIH who was somewhat critical or concerned about the Provenge vaccine. And one of the things he was concerned about was the cost. And he said that compared to chemotherapy, it was much more expensive. This article that came out today on the internet from Minionville, actually went through an analysis that I thought was fascinating because, in fact, if you compare the cost of chemotherapy, you have to include the side effects that come from chemotherapy. And the available therapy, which is a Taxol type of drug that was available, first of all, it didn't extend life nearly as much as the vaccine, but in addition, something like 25% of the patients who got that chemotherapy had severe side effects, so much so that they had to be hospitalized. So the cost of that kind of treatment and it has to include the cost of the complications and the side effects if you're going to do a comparison. But I thought it was a very interesting analysis, and I recommend it to you. Yes?
Yeah, and since most people, most men die with prostate cancer, but not necessarily of prostate cancer, again, how do you pick out a suitable candidate, and why wouldn't that candidate opt for surgical removal?
Oh, I'm sorry, that's a great question from another friend. I don't mean to suggest that this is a treatment that's used instead of surgical removal. In point of fact, most patients with prostate cancer choose to have their tumors removed either surgically or with radiation. This is a treatment that's reserved for patients who have advanced, otherwise essentially untreatable prostate cancer. So if you've had prostate cancer and you're cured, you wouldn't do this. No need. This is for patients who, however, have not been cured by conventional means. And almost all of them have tried conventional means. So not every patient, as was pointed out, with prostate cancer goes on to develop this kind of advanced disease that requires such therapy. In fact, it's a minority of the patients. I think there's 100,000 or so, or, I mean, I think there are a couple of million, if not more, patients with prostate cancer, but only a minority of them would be appropriate to treat with this treatment. Does that answer your question?
Well, again, why wouldn't surgical removal take care of the issue?
Oh, okay. So the question is, why doesn't surgery just cure everybody, right? Well, that's what the surgeons would probably want you to believe. Unfortunately, it doesn't work all the time because the tumor can spread. So if you can remove the tumor before it's spread to outside of what we call the capsule, then surgery can work very well, so can radiation. But if the tumor has spread outside of the capsule of the prostate into adjacent organs or other distant organs, it usually spreads in a manner that you cannot remove it surgically because it spreads in tiny micrometastases that are simply too numerous to be able to identify and remove surgically. So that's the problem with any tumor. Any solid tumor that has spread, it's almost no solid tumor can be treated surgically once it has metastasized. Al. So the question goes back, in a sense, it combines a number of questions, which is, well, why not treat patients with early stage tumors that are perhaps not widely spread yet, patients a little healthier? Wouldn't the treatment potentially cure such patients? And I'd say it's possible. However, if you can be sure that you're going to get a cure by a simple surgery, maybe that's to be preferred. So far, the data with, even in animals that are treated with vaccination alone, it's hard to cure tumors with single modality therapy. It's really hard. We're actually working on an approach that, in fact, my old collaborator, Dr. Strober, and I are working on an approach that combines tumor vaccination with a form of bone marrow transplantation that seems to have the potential to cure widespread tumors. we're very excited about that in mice so far. But no, I think it's going to take a really long time before that's shown, and you can imagine how long it would take to prove it. If this took 15 years to develop for patients who had advanced disease, can you imagine how long it would take to prove efficacy in patients who don't? So it'd be a big challenge. Yes, sir?
I have two questions, and one is logistical. You've run out of handouts. Can you make the slides available on the website?
Yes, I'm sorry about that. It was sort of a half-hearted way to make handouts because we didn't know that people would want them, but we can do that.
Yeah. You see me, I'll give you a business card and I'll see to it that you get.
Yeah, it'd be good to make the slides available, I agree.
And the second question is, what are the prospects for vaccines that are not individualized or ideally could be administered before the patient even gets cancer?
Okay, well there's two questions there. One was what is the possibility for vaccines that are not individualized, and the possibility for prophylactic vaccination against these other tumors. I mentioned that there's one approved prophylactic vaccine, and that's the one for cervical cancer. The challenge of prophylaxing or preventing tumors is that you'd have to identify antigens that are unique to the tumor or are involved in the cause of the tumor. I mentioned before that most tumors consist exclusively or almost exclusively of self-components, and presumably they're there because they serve a useful purpose, not just for the tumor, but for normal tissues. So there's a big challenge to that. If we discover that breast cancer is caused by a virus, and then we can vaccinate against the virus that causes it, and it should work. But until or unless such a discovery is made, it's going to be very difficult. The reason we can vaccinate against cervical cancer is not only because it's caused by a virus, but that virus is obviously foreign. So if we induce an immune response against the virus, it's not going to hurt your normal tissues. It's not clear that that would be the case for other tumors. Now, is it possible that by inducing an immune response against prostate acid phosphatase, you could prevent the formation of prostate tumors? I don't think so. I think that what you'd find is you'd find tumors that grow out that don't have prostate acid phosphatase because they don't need it. I think that the other key to the perfect vaccine is to find not just a molecule that's only expressed by the tumor, but that an antigen that is needed by the tumor for survival. So those are things that are, I think, pretty far off in the future. Now, your other question involved was, can you imagine a vaccine that would work for everybody, not just for an individual? And I think you can. The approach that I described so briefly in that last slide was to obtain molecules that can activate and target dendritic cells inside the body. So you don't have to remove the dendritic cells. Those molecules should work for anybody, as long as they're dendritic cell specific. You may have to bring the antigen to the dendritic cells, and that would be tumor specific, but it would work for everybody with the same tumor. Yes, Chris?
See a large percentage of this research being pushed offshore? I mean, the world's a small place, and there are a ton of pretty savvy companies, particularly in Asia.
Yeah, Dr. Gonzalez raises a good point, which is that given the cost of doing this kind of cell therapy in the U.S., is it likely that other countries might take up the slack? And I do believe that to be the case for cell therapies in general, because they're so expensive here and they're so aggressively regulated. I don't disagree. Yeah.
So you said that in effect what you're doing here is creating an autoimmune response. Do you see any tendency for the autoimmune response to attack healthy cells as opposed to tumor cells?
Yeah, so is there a risk that through this approach we're going to create autoimmune disease? And the answer is yes, absolutely unequivocally. That is the most significant concern with this kind of vaccination strategy. And if I were the FDA, I'd say we have to watch out for that. But my counter to that would be, is it not worth doing the experiment to find out? And what happens if you develop an autoimmune? Would you trade a terminal cancer for a treatable autoimmune disease? I would. But in the end, you're right. There's a trade-off, and that is the most significant risk. Now, in the case of prostate cancer, as I said before, who cares? But for other tumors, particularly those that involve organs that we need, it's a significant risk factor. Yes?
So is it known what the tumor does to eventually become resistant to the vaccine?
Well, in this particular case, the question was, do we know why the tumor can become resistant? In this particular case, we don't understand it at all. We do know from experimental systems how tumors can escape or evade an immune response. I implied one of them just a minute ago, because if you end up attacking an antigen, a molecule, so any cell that has that molecule would be killed. There are cells that don't have the molecule. The tumors are smart enough, if you will, and some of the tumors will simply lack the antigen because they're mutating, they're changing. And so unless the antigen that you attack is required for the tumor's survival, then eventually, theoretically, it can escape. So that would be an example of how that might happen. Yes, sir.
Is there any knowledge of this type of research advancing like AIDS research?
Well, that's a good question. The question was, can this kind of research ultimately prove helpful in AIDS research? And you may have noticed in one of my early slides, we actually did, years ago, an experiment, a clinical trial in patients who were HIV infected, where we isolated their dendritic cells, armed them with HIV antigens, and gave them back. It was perfectly safe. They generated some immune response to the antigens, but it didn't make any difference. Ultimately, we need an AIDS vaccine, not a treatment, but a prevention. And that has turned out to be really tough. Now, that's the subject of another hour's discussion. I used to give lectures on that subject, and I have to give one tomorrow morning, actually. And it's really fascinating and challenging as to how can we come up with a vaccine that would prevent a foreign virus. I just mentioned the idea that we can prevent cervical cancer by making a vaccine to human papillomavirus. Why can't we do the same thing with HIV? Well, it's not just HIV. We can't do it with hepatitis C either. They represent unique molecular challenges that I can discuss with you offline, but it's a long discussion. Yes?
This tumor antigens present within a primary tumor, metastatic tumor, any different from its respective stem cell tumor cell? And if so, are there any research trying to address the antigens in the culprit, the origin of the tumor?
Okay, that was a very sophisticated question, because there are lots of sophisticated people here. The question was, if I could rephrase, have you taken into consideration when you're in your vaccine design the cancer stem cell? Is that a fair way of saying that? So some of you may not have heard of the cancer stem cell theory, but it's actually an important theory, and it's one that a number of my colleagues here at Stanford have involved in generating, and there's some proof that it could be right. It's still controversial. And that idea is that when you have a tumor, you might have, let's say, 1,000 tumor cells. Only one of those cells, the cancer stem cell, is actually capable of causing the tumor. The others are bystander cells that grow fast, but they're really kind of dumb cells. The people that believe in the cancer stem cell theory believe that most of the chemotherapeutic approaches to treating cancer are all wrong, because they're designed to kill rapidly growing cells, but they don't kill the cancer stem cell. So it's a fascinating new area of research and investigation, and a very important one. And so the idea would be here that if we can isolate the cancer stem cell and characterize the antigens that it has, then you'd want your vaccine to be directed at it. And that is a potential aspect for the future. So I completely agree that that would be one of the reasons, in fact, it could be one of the reasons for tumor escape, is that we're not hitting the tumor stem cell. So a sophisticated question, and we're working on that. Yes, ma'am. So the question was, if I can rephrase, does the vaccine induce antibodies to the tumor? And if so, can you monitor the antibodies and thereby know whether the vaccine is working or not? Is that a fair way of rephrasing your question? Okay. In this particular case, There are antibodies that are formed, but it's not believed that antibodies are responsible for the effects of the vaccine. It's believed that the vaccine is actually activating the T lymphocytes to attack the tumor rather than antibodies to attack the tumor. Yes?
So you suggested that you might be doing some damage to the prostate, and I'm just wondering if that's been looked at, like how bad is that damage?
Yeah, so it gets back to this question of autoimmune disease of the prostate, and maybe I didn't mean to be misleading. In these trials, virtually all of the patients have had their normal prostates removed. These are, and I guess I didn't make that clear, but most patients, not all, most patients elect to have either surgery or radiation to destroy the prostate itself, not just the tumor cells, but the normal prostate gland. So it's not been possible to answer your question. Now, when this approach was used in rodents, in mice and rats, indeed it induced an acute inflammation of the normal prostate, an infiltration of white blood cells, an autoimmune, if you will, prostatitis. And that was one of the reasons why it was felt that the vaccine construct might work, is because it actually did this in normal rodents. Yes, ma'am?
What other cancers are in the pipeline for dendritic cell research, either by this company or elsewhere that you know of?
Okay, so the question was, what other cancer vaccines are in the pipeline with dendritic cells, either with this company or others? And I confess, I can't give you a definitive answer to that. Yikes. I know that Dendrion is actually using a very similar vaccine construct for bladder cancer, and they're just beginning those studies. And they had in their original pipeline dendritic cell-based vaccines for breast cancer and other tumors. I don't know, since I don't have a pipeline to Dendrion anymore, exactly what they're doing or what they're planning to do. As I mentioned before, using dendritic cells for these patient-specific vaccines is incredibly expensive. So most of the newer, or many of the newer approaches, not all of them, but many of them involve ways to try to activate the cells in vivo, using, for example, safe viral vectors to do that. And some of the most promising work involves the use of these recombinant vectors, be they viruses or other organisms that can be used to bring the antigens in to the dendritic cells and activate the dendritic cells. And I'm actually pretty optimistic about those outcomes. It's going to take another few years because they're just now beginning late-stage clinical trials. And they're in prostate cancer, breast cancer, probably colon cancer, I believe. And mostly these are companies that are pursuing this kind of work. The academics, we make the initial discoveries and then hopefully turn them over to companies to develop. Okay, we're getting near the end. Anything else before we close up? If not, thanks for coming, and hopefully we will.
Can you take questions? Are you going to stick around? Sure. Yeah.